Hi, this is Dr. Greg Jicha from the University of Kentucky Alzheimer's Disease Research Center, and I'm here today to talk to you about the SMART-HS trial, the first ever trial of targeted intervention for late. Our disclosures are below, and the current grant is funded by R01 AG 061111. So how did this all begin? The genetic linkage to HS aging was demonstrated by Dr. Pete Nelson and has been replicated in multiple cohorts, identifying a particular target gene, ABCC9. And this is really, really important. But the question to ask is, what does ABCC9 code for? Well, it codes for a potassium channel that is highly prevalent in the CNS. And this potassium channel can be regulated by an existing drug. It's not approved in the United States, but is used globally for cardiac angina and chest pain. And this improves blood flow to multiple organs. Nicorandil does indeed modulate this potassium channel as an agonist. So we know that this could be helpful on the basis of that genetic data, but what clinical data do we have? Well, we know that existing medications such as sulfonylureas used for diabetes treatment are antagonists rather than agonists of this channel, and that they themselves are associated with an increased odds ratio of dementia. We also know that Nicorandil has an extensive history of human subject use. It has low toxicity and potential beneficial impacts, including those on the brain and in relation to all-cause mortality, as shown in the lower right-hand graph from a study from Japan. So there are some considerations. This is an agent that leads to vasodilatation, and so vasoactive nitrates are excluded among other medications. So the study itself is designed as a phase 2A proof of concept. Our study outcome measures are safety always as a primary outcome, but our secondary outcomes include efficacy measures, including imaging, hippocampal volumes, as well as cognitive measures focusing on memory tasks. Exploratory outcomes are abundant, and we hope to use the data collected in this clinical trial to further our work in search of specific biomarkers for TDP-43 pathology and late. So the study design itself is a 96-week drug exposure, approximately 60 participants randomized 1-to-1, annual MRI, CSF at screening and end of study, of course, biomarker blood assessments collected all the way through, as well as other safety assessments and cognitive functioning. So how are we going to ensure that our patients actually have late? We don't have an antemortem biomarker, and that's critically important. And we know that multiple pathologies can contribute to dementia. But if we look at this and start to exclude those at age less than 75, potentially eliminating many with FTLD, if we exclude those with Parkinsonian symptoms and psychosis, we get rid of the alpha-synuclein. If we exclude those with positive p-tau, if we exclude those with abnormal A-beta, and if we exclude those with significant cerebrovascular disease, what we're left with is TDP-43, the target for our intervention. So we've looked at this in great detail and powered for both safety as well as secondary efficacy outcomes, and the study is moving forward currently. We also hope, as I alluded to previously, to use the biological fluids, the imaging collected as part of this clinical trial to further biomarker exploration. And we do indeed think that we've identified an HS pattern from routine AD biomarkers, but more importantly, looking for specific TDP-43 HS aging biomarkers, and we do have some candidates, as you can see in the right lower portion of the screen. So this really is an exciting time for us. We're actively recruiting subjects. This study represents the first human clinical trial of a therapeutic agent specifically targeting late. So while the disease is just being debated in the field, we are already moving forward towards therapeutic interventions. We think that we can enrich for late using exclusionary criteria from both the clinical and antemortem biomarkers developed for other disease states. And we're going to leverage as well, because what we really need in the field is specific antemortem biomarkers for late. So I'd like to thank my collaborators, Dr. Nelson, our NIH funding, the ADRC and the clinical trials unit, but most importantly, the patients that not only engage in our clinical trial activities, but are also willing to sign an autopsy donation and consent form so that we can move these valuable discoveries forward.